Hi, everyone. Our speaker today is Brian Kraus, a graduate student in Dr. Katerina GRATTON's lab, who we heard from a few weeks ago. Brian completed his undergrad degree at SUNY Buffalo and his master's in clinical psych at Ball State University. From there, he went on to work at the University of Michigan. And now as a grad student, he focuses on the neural correlates of individual differences in behavior, working with both EEG and fMRI to explore topics like creativity, culture, and psychopathology. Just this year, he first authored this cool paper on variance similarity and task and resting state data, which he's going to review during his talk today. Thanks again, Brian, for joining us, and we're super excited to hear your talk.
Awesome. Thank you so much for that wonderful introduction. My name is Brian Kraus. We'll just reiterate all the information that was just said now. I'm from Katerina GRATTON's lab here at the Department of Psychology at Northwestern University. And very excited today to be giving this talk at The Ohio State University. So this talk today is going to be on what our lab calls network variance and their stability across different cognitive states and tasks. And so just to give you a brief outline of the talk I'm going to be presenting today. So I'm going to at first go over how there's a trait-like stability to these network variants. So I'm going to start off by giving you some background on functional connectivity, as well as what we call network variance, and also go over some of the evidence for stability of network variance across different task states from a recent study we just did. And then I'm also going to talk about some ongoing work on the possible clinical relevance of network variance. So I'm going to briefly review some evidence that network variance might have relevance for clinical psychopathology, and then also discuss a little bit about a current ongoing project we have on the relationship of internalizing disorders to network variance. So I'm going to start off with this background on functional connectivity and network variance. So I think this will be review for most people at this talk, but our lab focuses a lot on functional connectivity. So you can imagine, for instance, that if you put someone's brain in an MRI scanner, probably still attached to a person, but inside the scanner, you can have them perform a resting state where they sit in there and do basically nothing. And over time, you'll record signal from many voxels that compose this different brain here. And you can imagine you'll have a couple different ROIs you could choose. So for instance, you could choose one in the left or right motor cortices represented by these lovely circles here. And you could average the time series of all the voxels within these ROIs, and you could correlate them. And in this instance, you would get a pretty high correlation between the two of them because at rest, the left and right motor cortices are functionally connected. However, you could also choose a different ROI, such as the left visual cortex. And if you try to do a similar correlation with the motor cortex from this region, what you'd find is that they have a pretty low correlation because the motor cortex and visual cortex are not functionally connected at rest. And that's sort of represented in this correlation matrix over here, where you can see that the motor cortices are very highly correlated to each other, but not to the visual cortices. And another way of representing this functional connectivity is through a functional connectivity matrix, where, for instance, you could have either voxels or ROIs, and you could correlate the time series of these voxels or regions of interest to each other. And by doing that, you could represent how every single voxel or region of interest in the brain is correlated to every other region of interest or voxel. And using this type of representation of functional connectivity, you can also use it to estimate what we call functional networks. So these correlations among brain regions can be clustered into a reproducible group network structure. So here are two examples that are pretty famous. One from Jonathan Power when he was at WashU St. Louis, another one from Thomas Yeo when he was at Harvard. And even though both of these studies use very different samples of participants and very different methods of actually clustering the different brain areas into networks, you can see that they have very similar spatial topography. So you can see here this bright red that Jonathan Power used for the DMN and this kind of pinkish red that Thomas Yeo used. You can see that you get very similar spatial topographies of these networks, and the same is true for the frontal parietal network and singular opercular networks, both of which are involved in cognitive control. However, though, what our lab's really interested in are individual differences in functional connectivity. And so far, in every individual we've looked at, we find these consistent variations from the group network structure in functional connectivity. So just to give you an example here, so this would be an example of a group level network structure up here that I just showed you. So this is going to be one that's very similar to the Jonathan Power one that I showed just before. And you can also compare that to networks of individual subjects. And as you can see here, you can have an example on the left here where in the group network, this region of the dorsolateral prefrontal cortex is usually occupied by the singular opercular network. However, in this one person, MSC06, who's one subject, you can see here that that region's been kind of expanded. The frontal parietal network's kind of expanded to take over some additional real estate that it doesn't normally have in the group level. And alternatively, you can also have another situation where in the group network, you have a region such as this DLPFC region here that's pretty standardly in the frontal parietal cortex. However, in any given individual, you could have a little patch of here default mode network, which kind of appears in there, which is kind of idiosyncratic and very different from functional connectivity that you observe within the group. And so our lab's interested in studying these variations. And one way that we do that is through what we call network variance. And so network variance we define as regions of an individual's functional connectivity that greatly differ from a group average. And so just to give you a little bit of background on kind of the theory of how we sort of interpret what network variants are, you could imagine a situation where you have a typical frontal eye field in an individual. So this would be like a group average frontal eye field that you estimate. And so this region would be composed of different populations of neurons, such as those related to saccading, visual processing, or attention. I know this is kind of a oversimplification of the cytoarchitecture of this area. I think it's still a really interesting example to look at. And you can imagine kind of an example like what I just showed you with the frontal parietal network or singular opercular network, where this region is present in individuals, but for whatever reason, it's just slightly smaller or slightly bigger than you would see in a group average. You could have a contraction or expansion. In a similar vein, you could also have that region be displaced slightly. So it's about the same size as normal, but for whatever reason, it's moved several centimeters across the cortex. Or you could also have a situation that's more akin to the other one I presented, where the default mode network kind of pops into the middle of the frontal parietal network, and you have this kind of unique region that sort of isn't typically present in group averages and doesn't totally have a homologous comparison across people, but still seems to associate with a known functional network. And what we have tried to look at in our lab is how stable these individual differences are, such as network variance. And so some early evidence from previous studies from Washington University, St. Louis have shown that functional connectivity is really reliable, or also trait-like, you could say, with enough data. And so this right here is essentially showing you a split half correlation. And this data comes from Russ Poldrak's My Connectome dataset. So for those of you that aren't familiar, about 10 years ago almost now, Russ Poldrak had the idea to scan himself 3 to 4 times a week in an MRI scanner every week. And essentially when he did that, he got, hours and hours of resting state data and some task data on his own brain. And at WashU, they took that and essentially decided, well, how similar is this functional connectivity within this one individual over time? So what this is showing essentially is that you can imagine that there's 800 minutes total that came from that entire scan over the course of the year. So if you hold out 400 minutes and use that to create a functional connectivity matrix, and then you slowly add 5 minutes at a time to this other functional connectivity matrix, how long until you get enough data to where it's pretty reliable, where some amount of data from this split half will equal the 400 minutes from the other split half? And what they found is something that we found across several other samples, is that around 40 minutes, you start to get really reliable data. So this is above a 0.9 correlation to the other half. And especially around 100 minutes, it really starts to asymptote, and you end up with a value that's here between 0.95 and 1, but I would say in other data sets, we typically end up maybe around 0.9 or so. And so not only are these variants reliable in a split half sort of sense, but they're also pretty reliable across time. And so this map here, I'm going to try to orient you a bit to this spatial map. I'm going to go into a lot more detail about how this is estimated in a few more slides. But just to try to give you a quick kind of rundown of what's happening here. So what I'm showing you are 10 sessions from one subject. MSC02 is from a Midnight Scan Club data set. And essentially, what we did here was try to compare individual functional connectivity to group functional connectivity. So what we did was we obtained a spatial correlation of each individual compared to the group. And you can see here, this is the spatial correlation. So most of the brain you can see is pretty red. So a lot of individual functional connectivity is pretty similar to what you observe in the group. So people don't usually drastically vary from what a group functional connectivity estimate would show, but they have some regions that have a pretty low spatial correlation to the group functional connectivity estimates that we obtain. And as you can see here, across all 10 sessions, those regions tend to be pretty stable. So this is over the course of a few weeks, and you can see that at each session, there's about a half an hour of data sampled, and you get pretty reliable spatial differences that replicate. And when you look at within subjects here, you can see that you get a pretty high interclass correlation for how similar MSCO4, for instance, is between one session and their other sessions. And it's much higher than you get when you randomly shuffle across subjects. So for instance, how similar is session 6 of MSCO7 to session 6 of MSCO5? You can see those values are all here in these random between subject permutations, but they're nowhere near as high as the within subject data. And so while we have some evidence that variants are pretty temporally stable and show good split half reliability, we don't really have great evidence yet, before this paper at least, for how stable they are across states. So we know that whole brain functional connectivity is pretty stable across states though. So this is a paper my advisor published in Neuron several years ago. And essentially what she found was that in trying to look at the state effects, so you can see here, these are correlation matrices with each person from the Midnight Scan Club dataset, which I'll go into a bit more detail about in a couple minutes. And you can see here that each person is very highly correlated to themselves and sort of maybe mildly correlated to other people, but not too correlated. But another thing that this graph is supposed to show is that down here, it might be a little bit hard to see on your screens, but there is a little boxes here above this name, and they all represent different task states. So there's a resting state, a coherence task, a semantic task, a motor task, and a memory task. And if these were all very correlated to each other, you would expect to see higher correlation specifically along these dimensions here. So where these are represented, you should see higher correlations within a subject that are along these different task blocks here. And as you can see, you don't really see that. You don't really see a vertical pattern here of high correlations that go down within each subject. You just kind of see that each subject is very similar to themselves, regardless of what task state they're in. And to try to put numbers to this, in this paper as well, what my advisor did was look at how different aspects of functional connectivity attributed to how much variance they attributed to network similarity. And you can see here that a large part of network similarity is that there's a group estimate of functional connectivity that many individuals are somewhat similar to. Individuals are very similar to themselves in functional connectivity above and beyond the group. There's a little bit of a group task effect. So this basically means the memory task accounts for a little bit of variance across people. But the third biggest source of variance here is individual by task. So for instance, the motor task is sort of consistently altering functional connectivity within individuals. But you can see that if you added these two task portions up, you'd get to about this dot, which is about 0.8. So you can see about 20% or so of the variance here is accounted for by task differences in functional connectivity. So we wanted to investigate if that would be true for network variance, or if network variance would show additional state effects that aren't typically seen in the whole brain. So I'm going to stop here and ask if anybody has any questions so far on the background or about network variance so far. Okay, so we'll move on to the study here. So in this study, sort of based on what I just said, you could imagine several alternative hypotheses. So one is that network variants show this extreme deviance from group functional connectivity estimates because they're more susceptible to transient effects, such as ongoing cognition. So for instance, if you're in a resting state in a scanner and you're just thinking about things, maybe these areas just have additional intra-individual variation. And it's also possible that network variants show high stability across different task states. And this would support that they have a trait-like invariance to changes in cognitive states. And so the trait-like invariance thing is something that's important because, especially if you're thinking about network variance as potentially being useful as something like biomarkers, you would ideally want something that's pretty invariant across time and state so that you could measure it at one point and, months later, or even, if somebody was thinking about something else, for instance, during a resting state, this functional connectivity associated with network variance wouldn't change very much. It would be pretty stable. So it would be a stable trait-like marker of some personality trait or something like that, or intelligence that you could measure at a later time point. And so in this study, we use what's called the Midnight Scan Club dataset. So there's 10 participants that were sampled in this dataset. One is not reported here because they had a lot of head motion in the scanner, and head motion just wreaks havoc on functional connectivity estimates. So we do not include them in this study. And this data set includes 10 sessions. So there's a resting state, each session that's 30 minutes long, the human connectome project motor task where you have to do things, let's say like move your fingers, move your tongue, move your feet, things like that. There's also these semantic and coherence tasks, which I will mostly refer to as the mixed task. So I'll kind of be analyzing that together throughout this talk here. And also a semantic memory task. And so just to give you just general methods here that I think will be relevant for the rest of the talk. So when we look at differences between task and rest states in our lab, we always put the task model through a general linear model first with a FIR basis function. And the reason for that is that we don't want to get spurious functional connectivity from regions which have a HRF that occurs in response to a stimulus. So for instance, you could imagine a situation where someone's watching a movie and there's a visual stimulus and an auditory stimulus, and both of those stimuli will create some version of an HRF. And then as a byproduct of that, the auditory and visual cortices will look more correlated to each other than they actually are. And so to try to get at the kind of intrinsic activity in the brain during the test, we try to fit a general linear model to the data and do our best to try to account for those signals before we do these functional connectivity analyses. And we also do global signal regression, and we censor high motion frames. So any frames which have a frame-wise displacement of greater than 0.2 millimeters, we censor and remove from analysis. And for all of these analyses I'm going to be reporting, except for one, which I'll specifically mention that this isn't true for, what we do with the task data is that we combine the data from different tasks and try to match it in length to the resting state data across participants. So for instance, we'll take, if somebody has 40 good minutes of resting state data across a few sessions we combine across, we'll say, okay, let's try to get 13.3 minutes of motor, 13.3 minutes of mixed task, 13.3 minutes of semantic task, and concatenate all those together. In some cases, we didn't have quite enough, so we had to kind of oversample from some tasks to get to that amount, but we tried as best as we could to equally sample from the tasks. And also, we mapped the data from the volume to the surface, which isn't that important for many things I'm talking about, but I will be using the term vertices to refer to essentially those are voxels that have been mapped to the surface. So just in case that term's unfamiliar with you, a vertex is basically just a surface voxel. And so now I'll go into how we actually estimate network variance. So basically what we do here is we get seed maps for every single vertex in the brain here. So you can see here, these are seed maps for individuals. So here's MSCO2 and MSCO6. And we also do the same thing for a group seed map. So this is what we call the WashU 120. So this is resting state data from 120 people that's been concatenated together, and functional connectivity estimates have been obtained using that. And so essentially what network variants are is that you can see, for instance, here in MSCO6, I have this little, I'm not sure if you can see, it's kind of small, but there's a little white dot here inside this black circle, and it's a little seed region in here inside the inferior parietal lobe. And this is a node of the default mode network, and consistent with that in MSCO2, you can see that there's positive correlations with the precuneus and cuneus and medial prefrontal cortex, other common nodes of the default mode network. And that's consistent with the group seed map. So same thing here, same seed in the inferior parietal lobe. We get the cuneus, precuneus, medial prefrontal cortex, positive correlations. However, when we go up to MSCO2, I have the same exact seed region. But what you'll see here is that the connectivity pattern looks very different. So we have negative correlations in the precuneus and medial prefrontal cortex, which is sort of the opposite of what you'd expect. So it looks like there's a pretty significant difference in functional connectivity between this particular area and MSCO2 and what you typically expect in the group. So the way we quantify that is we do what's called a spatial correlation. So essentially what we do is we will take all the correlations between this area and every other region of the brain, and we kind of just string them out in a big line. And we do the same thing for the group average, and then we just run a correlation. So for instance, we say how similar are the correlation patterns of MsCO2 to the WashU 120. And what you can see here is that we get a very low correlation to the group for MsCO2. So we have this region over here that doesn't look very similar to the group, while in MsCO6, we get a very high correlation because this looks pretty default mode networky over here. And so as it would stand, that shows a pretty similarity to what you'd expect in the group average. So once we obtain these spatial correlation maps, which give us a good idea of where functional connectivity in individuals actually differs from a typical group average, then what we do is threshold it at the lowest 5%. So then we get the lowest 5% of these blue regions essentially, so the lower end of this tail. And after that, what we do is we take the seed maps for all the vertices in this kind of blue blob here that gets thresholded, and we compare that to typical group average networks. And we say, okay, we don't think this looks very much like a default mode network node. What node do we really think this network is a part of? And so when we did that for this particular variant, what we found is that, this seed actually looks a lot more like something you'd see in the singular opercular network, not really like something you'd see in the default mode network. So here we've assigned this region to be part of the singular opercular network and not the default mode network. And then down here, you'll see for MsCO6, this region with very similar does not end up getting thresholded. And so there's a region below it you'll see which gets assigned to the default mode network, this little red blob right here, but this particular seed I've been talking about is not a network variant in this person. So once we did that, now we were ready to move on to some of the results from this paper. So the first thing we wanted to just show is that similar to other data sets that we've looked at, we get a very high reliability of these spatial correlation maps, which we use to estimate network variance from. So at first we just started with resting state data just to show that we would get what we've seen with other resting state examinations of this. So you can see here that in a similar split half analysis to what I talked about earlier, you hold 100 minutes out, you slowly add in 5 minutes until you get up to 100 minutes. And after about 40 minutes, almost everyone's above 0.8. And especially as you get more data, people end up being at about 0.9. You'll see some people don't have quite enough data, so they don't get all the way up to 100 minutes. And this right here is a measure of variant spatial location reliability. So this right here is a bit noisier than the continuous spatial correlation map because at each value here, we have to threshold the lowest 5%, and just some noise in the values can kind of change a bit how stable those are. But you'll see that after about 70 minutes or so, you still start to get above 0.7, fairly stable spatial location reliability. And so by comparison here, we have our task data. So we get a similar reliability curve here for the spatial correlation maps, where 40 minutes, almost everything's above 0.8 and almost above 0.9 by 100 minutes. And similar here with task reliability as well. So after about 70 minutes, most people are above about 0.7. There's a couple below, but even they eventually get up to 0.7 with enough data. So we feel pretty confident here that we were getting reliable individual level functional connectivity from both task and rest data. And then we were curious how similar the network variants were that we defined were across states. So just to kind of give you an idea of what these maps look like in a spatial correlation map. So these are maps from MsCO2 and MsCO6. And here we have maps which we've defined with resting state data and here with task data. And these are totally equivalent on the amount of data. So both subjects have the same amount of data in task and rest. And they're also equivalent to each other. So all these subjects have the same amount of data. And so you'll see here, similar to MsCO2, this region that I was talking about earlier is there in rest and task. And you see that a lot of the regions you see are pretty stable across the two. But there are some regions where you can notice some differences. So for instance, here in the medial prefrontal cortex, you'll see this area right here looks pretty dissimilar in rest. But if you look at it in task, it doesn't look really quite as dissimilar. It looks kind of more middle of the road functional connectivity. And you can see similar areas down here too for MsCO6 if you look around. And essentially what we wanted to do then was say, okay, what about if we do a spatial overlap map here? How spatially similar are these? So here is just kind of a visual representation of the analysis we did. So we essentially said, okay, we define vertices in task and rest. And as I said before, the thresholding does add a little bit of noise to it. As you can see in rest, this area is considered a variant only in rest and not in task, even though they both have pretty low correlations to the group. And you'll see here, though, that even though there is a little bit of noise from this thresholding, there's predominantly most of the areas overlap, which is represented by this red between the two states. And there are also some which are only present in tasks, which are represented by these blue areas here. And so to try to put numbers to this comparison, we ran an analysis using this essentially this exact map here, only we just binarized it, and we ran what's called a Dice-Sorensen correlation. So a Dice-Sorensen correlation is just a way of correlating binary values. So essentially what you would do is you would set all the values in this spatial map that were variance and rest to 1. You do the same thing for task, and then you'd run a binary correlation between the two and see how overlapping they were. So as you can see here, we have some within state comparisons. So essentially what we do is we have split halves of the data. So we split the rest and task in half. So one of these green dots here will represent rest to rest correlations. The other green dot will represent task to task correlations. And the blue dots will be between states. So each blue dot will be one of the rest split halves to the other task split half. And what you can see here is that there is, across all individuals, there's a pretty consistent pattern where within state comparisons are slightly higher than between state. So you can see that over here. You can see that within state, the dice correlation mean is about 0.66. Between state, it's about 0.55. And however, though, if you look at this black dots down here, these across subject comparisons, what you can see is these are permutations where essentially I asked, well, how similar is MSCO1's task split half to MSCO3's rest split half? And that's what these black dots down here represent. So you can see that although there are significant differences here between within and between states, indicating that there are some state effects, they are much, much smaller than between subject effects. So if you were trying to estimate network variance from task or rest data within a subject, you'd be much closer to the truth than you ever would be by looking at what you would expect kind of at random or between subjects. And the effect size between these two is, this is like 5 standard deviations, and this is more closer to 2 standard deviations. So it's a much larger effect size between these two than you would see between these two different states. And we also wanted to look at network assignments. So just to kind of review how we do network assignments here, I touched on this a bit earlier, but essentially what we do is we take areas that have a low correlation to the brain and threshold, or low correlation to the group, sorry, and threshold them. And then what we do is we compare the connectivity of these contiguous regions to our group templates of functional networks. So we take our group average and we try to look and see what network template is most similar in a winner take all fashion. And so these purple ones represent singular percular, for instance, red represent default mode network. And this one right here would be a visual one in the back here on MSCO2. And so essentially what we wanted to see was how similar are these assignments for network variance across individuals. And when we looked at rest and task variance, what we see here is that you get a very similar number of variants across both states when you define them across everyone. So this is showing you how many variants are in, for instance, red is default mode network, or yellow is frontal parietal. This one right here is singular percular, this purple one. You can see that the distribution of variance is relatively similar across the two states, and you also get a similar total number of variants, though there are some differences. For instance, there's slightly less frontal parietal variance in task than rest, but they do come out to be pretty similar. And here we tried to look at the dice correlations for network assignments. So here's essentially what we did is we would go into, in the rest variance, for instance, we would go into rest and we'd say, okay, I'm going to take all of these vertices in task, and I'm going to say, how do these assign to the same network? And obviously in this case they do, because that variant exists almost exactly here. But you could have a situation like this one up here, for instance, this default mode one here in the prefrontal cortex. You might, we would do the same thing here and take these regions that aren't part of a variant, or these vertices that aren't part of a variant, and we would also assign them to a network and see if they both came out as default mode network. And what you can see here is that you end up with an average of about 0.7, 0.8, or 0.8 more like in dice correlation. So that's the amount of times that they assign to the same network. So you can see that this is also pretty stable. And you'll see that MSCO3 has a little bit lower values here. They had a low number of variants overall. They only had like 3 or 4 in tasks. So this is probably reflecting kind of noise. And just to show you that This is most likely noise from just kind of thresholding operation here. What we also did was we looked at the seed map correlation. So essentially what this is showing you is if you took those areas like I just explained and took the same vertices and looked at the seed maps across task and rest, so how the correlation of those vertices to every other vertex in the surface, what you would see is that you get a very high spatial correlation for the Pearson correlation. So this is essentially showing that in a continuous estimate, which is a bit less noisy than the binary estimate I was just showing you, we get very high similarity between states. And these black dots are what would be expected across subjects. So for instance, how well would those seed maps between MSCO3 and MSCO9 correlate? And it's not very well. It's kind of the mean of 0 for all of them. And another thing we were interested in looking at is how network variance compared to the rest of the connectome. So what we wanted to see here essentially was what we were interested in our data set is looking at the very least similar vertices. So here what I've done, essentially with the way we define network variance is we take the lowest 5%, so that would be 0 to 5%. So instead what I did here was I broke this up into a different bin. So I thresholded, okay, I want the least similar to the group. So 0 to 2.5% similarity. Then I could get 2.5 to 5%, 5 to 7.5, 7.5 to 10, 10 to 12.5, and then 12 to 15% here. And I'm only showing you a subset of them because it would look like ants on a, if I put them all up there. But essentially what's really interesting about this is if you look at the least similar and most similar to the group, you can see that on the tail ends of the distribution, you actually get these contiguous parcels, whereas when you kind of look in the middle of the distribution, it almost looks like someone sprinkled sawdust on the brain. You don't really get these very consistent contiguous regions. And so we wanted to see is the stability of network variance or these regions that are in the very tail end of the distribution, are they more stable than these kind of regions in the middle of the distribution that have like an average similarity to the group? And what we actually found was just that. So this is essentially showing you those bins I just showed you visually. So you go from 0 to 2.5, 2.5 to 5, 5 to 7.5, 7.5 to 10, and so on. That's what each bend of this curve is. And you can see that on the very low end of the similarity of the group, we get really high stability within states and between states. And the only other place we really see any stability is on the total opposite spectrum, where things are very similar to the group. And another thing that's interesting to note here is that we do the same thing across subjects, and we find that essentially there's very low similarity between subjects in the amount of overlap in these regions that we call variance, but there's actually a bit more overlap in the regions that are very similar to the group, almost indicating that subjects tend to have similarity to the group in similar regions, but not really at this end of the distribution. And another thing we tried to look at here were task-specific effects on network variance. So up until now, I've been presenting data where I've combined all the data from these tasks together. But here we tried to split it out and say, okay, how similar are variance and rest compared to these individual tasks? And I will just note that this, unfortunately, we don't have a ton of data for these tasks compared to when we can combine them all together. So we don't have like 40 plus minutes of data per person per task to do this sort of comparison. So I am not totally, I wouldn't say that these like are definitive results. I mean, there's no task specific effects on functional connectivity or anything like that, but we tried to do the best we could here. And so this is essentially what I've been showing you, which is all tasks combined. And then we broke it out by memory task, semantic coherence task, which I've been calling the mixed task, and the motor task. And as you can see here, what we're really interested in is this rest to task. So how similar is the variant spatial overlap between resting state data and task data that's matched in time? And as you can see here, there's not really a big difference. And if you run a ANOVA on this, you don't get any significant differences for rest to task. So we don't find any task specific effects on network variance and where their locations appear. So just to kind of summarize what I've gone over so far. So network variants are largely trait-like, and they tend to occur in similar locations and show similar connectivity profiles across states. So this is more consistent with the second hypothesis I presented earlier. However, there's definitely some susceptibility to states as indicated by the fact that within states, you do a little bit better than between states, but not a lot. And we only saw evidence here for a monolithic state effect, so like a pan task effect, but we didn't really see any effects for specific tasks. And some considerations for identifying network variance. So they tend to be less similar across states than within states. So they're about 17% less reliable. So that was the chart I showed, which essentially the green within state comparison was slightly larger than the blue within state comparison, or blue between state comparison, I'm sorry. And however, though, if you compare this effect to having a smaller amount of data, for instance, if you tried to make network variance with like 5 minutes of data versus 40 minutes of data, you'd actually be about 56% lower. So this having a lesser amount of data is a lot worse than trying to combine data across states. So if you said, oh, I only have 10 minutes of resting state data in my data set, but maybe that's better to estimate network variance from than adding this half an hour of task because there's these state differences. We don't really find much evidence for that. It seems like having more data is definitely better than trying to estimate it with small amounts of data, even if they're not from the same state. So to us, this supports the idea that task data can be used to identify network variance. And so I'm going to just pause here and to see if anyone has any questions about any of that I can clear up real quick before I keep going.
I have something I want to see if I can clarify. So you show the pie charts of where variance was most likely to occur. I think like within each participant.
This right here?
Yeah. And so it looks like the default mode network and the visual networks are the most likely to have variance. Am I interpreting that correctly?
So what this is, so this is showing essentially like if I added across all the individuals in the MSC and I said like, okay, this person has 1, 2, 3, 4 default mode networks. And I was like, okay, I'll add those 4 in. And then I went to MSCO2 and I said, okay, 5, 6. And this is like a number of counted variants across everybody. And so it would be showing here that default mode and singular opercular would have the 2 most, and visuals, this little blue slice right. Sorry, some of these colors aren't super distinct.
Oh yeah, the color I missed up. Okay.
And so that's what this would be showing here. So sorry if I was unclear about that.
Okay, yeah, because I was just wondering, as you were talking about this, both, I guess, spatially and functionally, if you've been identifying where is there, which parts of the brain isn't most likely to differ across individuals. And I was wondering if these graphs suggest that, well, in default mode network seems to be a big area, which I think theoretically would be plausible because it's associated a lot with individual differences and sort of our conscious states and that's and self-awareness and that sort of thing.
Yeah, that's a great question. And I'm gonna, in the next part of the talk here, I'm gonna get into that a little bit more about which regions of the brain vary. But yeah, default mode network is definitely one of them. So what this is technically showing, though, just to clarify, because this is confusing and I didn't say this, so I'm sorry. I should have been clear about this. So what this is saying is like where other regions aside from the default mode network are more likely. So to be a default mode network variant, one of our requirements for estimating it is that when we compare it to the group average, it can't overlap more than, I think it's 50% off the top of my head with the group estimate of the default mode network. So this has to occur in another network. So it has to occur in like the frontal parietal network or the singular opercular or something to be a default mode network variant. So what this is showing is that like other regions of the brain tend to show network connectivity patterns similar to the default mode network, but those aren't normally in the group default mode network. But I think you're onto something really good there with which regions of the brain show this, and I'm going to get into that a bit more in the next part of the talk.
Okay, perfect. Thank you for clarifying.
Yeah, of course. Thank you.
Brian, I have a question about some of the correlations, just because I'm doing some reliability stuff myself. So this is very interesting to me. So for all your Pearson correlations with the seed stuff, I'm just making sure I'm getting this right. You're turning the connectivity matrices into vectors, and then you're just running the Pearson correlation between those 2 vectors of the same matrix, right? Or matrices from different people?
Yes. So essentially, these aren't, what I'm showing here is technically between it would be vectorizing one of these spatial correlation maps.
Mhm.
We've also done that for actual functional connectivity matrices in the similar way you're describing, yes.
Okay, and then could you remind me, you said that the Dice-Sorensen correlations are binary ways of comparing. Could you just explain that one more time for me?
Sure. So essentially what we do is, right, like we would threshold our spatial correlation maps and we would get the lowest 5%. And then what I would do is if I wanted to say, okay, now that I sort of have a binarized template for task and rest, now if I stretch those out into vectors, how similar are the binary correlations essentially between those? So how often do I get ones that occur in the same place across these two vectors versus 0, 1 mismatches essentially?
So your general structure for doing split half reliability or ICC or Dice or Pearson is basically the same. You're turning everything into these vectors and then across the vectors, correct?
Yes, correct.
Okay, thank you.
Thank you.
Hey, Brian, very quickly. So your comparisons for the group are done within the group of 9 subjects, correct?
So the group average does not include these 9 subjects. So it's an entirely separate group called the WashU 120, which was collected at some point years before the Midnight Scan Club ever was conceived. So these are from previous WashU data sets.
Gotcha.
Okay. So sorry, I gotta skip back through here. All right. So now I'm going to talk about some evidence for possible clinical relevance of network variance. So I'm going to start off by taking a brief foray here into major depressive disorder and just very quickly go through here that each year, estimated 3% of the world's population experiences an episode of major depressive disorder, and nearly 1 in 5 people will experience an episode of MDD in their lifetime. There's also large psychosocial and economic consequences for major depressive disorder, not just for the people diagnosed with it, but also for loved ones and others who sort of experience their distress with them. And so a diagnosis of major depressive disorder is defined by experiencing 5 of 8 symptoms during any 2-week period. So there's cardinal symptoms, which you must meet at least one of, such as the depressed mood most of the day, nearly every day, markedly diminished interest and pleasure in all or almost all activities, or also known as anhedonia, significant weight loss or weight gain, slowing down of thought and reduction of physical movement, fatigue or loss of energy nearly every day, feelings of worthlessness or excessive or inappropriate guilt nearly every day, diminished ability to think or concentrate, or recurrent thoughts of death or suicide. And despite the fact that many people are diagnosed with major depressive disorder, there's actually many differences among people who are diagnosed with it. And in fact, if you do the math, there's actually 227 unique permutations of symptoms which can constitute a diagnosis of major depressive disorder. And this isn't just sort of an academic exercise of computing how many possible permutations there are, but actually when people present to a clinic, there's actually a huge heterogeneity in the actual symptoms they present with. So this was a study by Zimmerman et al. With 1,566 people. And essentially what they find is that there are 9 different symptom combinations, which account for about 40% of the diagnosis. And then after those 9, there's 161 other ones that kind of fill up the rest of this slice of the pie here, and they all account for the other 60%. So there's a lot of heterogeneity in the different symptoms that people present with. And there's also similarly heterogeneous evidence for disrupted functional connectivity in major depressive disorder. And so major depressive disorder is related to disruptions in large-scale brain networks versus single regions. So here you can see one meta-analysis by Mulders et al. In 2015, and the same year, another one from Kaiser et al. And while there's some agreement between these 2 meta-analyses, for instance, hyperconnectivity within the default mode network is found by Kaiser as well as Mulders. There's also some areas of pretty severe disagreement. So Kaiser says that there's hyperconnectivity between the frontal parietal network and the default mode network. And the most similar network in Mulders' review is the central executive network, and he actually finds decreased functional connectivity between the default mode network and central executive. So there's a lot of disagreement here as well about what the functional connectivity correlates are of MDD, and this probably has a lot to do with the heterogeneity in symptoms I just mentioned earlier, but also heterogeneity in individual functional connectivity. So In humans, network variants occur primarily in association cortices. So these are the spatial locations of network variants. So you can see here that the Midnight Scan Club, which is what I just went over earlier, we have lots of variants in the dorsolateral prefrontal cortex and temporal parietal junction. You can see a similar thing in the Human Connectome project as well. We have temporal parietal junction, dorsolateral prefrontal cortex, and also some areas of the medial prefrontal cortex and cuneus. And as was mentioned earlier, a lot of these are within the default mode network. For the inferior parietal lobe is right around here, the medial prefrontal cortex and precuneus, and also this one up here is part of the frontal parietal network. So a lot of higher order networks are implicated and tend to be in regions where network variants occur, and they tend to occur pretty rarely in unimodal cortices, such as the visual cortex or the somatomotor cortex. And there's some initial evidence as well that these individual differences in network topography correlate with behavior. So just to kind of review what I just showed here, this is sort of where variants tend to occur. There's a study from last year from Ted Satterwhite's group that shows that essentially the amount of spatial territory that's taken up by the frontal parietal network is very associated with executive function performance. So for instance, here, if you have this variation in the area that's taken up by a certain brain network, that alone can account for differences in behavior, not even taking into account actual functional connectivity strength between the regions. And network variants themselves have also been associated with clinically relevant variables. So we have here, we have a sort of a finding from Ben Seitzman's paper showing that the default mode, people who have more default mode network variants, so more variants that are assigned to the default mode network, not variants that occur within the default mode network, tend to report lower positive life experiences, which is a risk factor for depression. In addition to that, they also tend to report a higher history of drug abuse. So that's also something that's comorbid with many psychiatric disorders. And so if anyone has any questions now, I can take questions on that. Otherwise, I'll go on to a current project and just briefly discuss something we're doing now on the relationship of internalizing disorders and network variants. All right, so in this ongoing project, we're collaborating with Dr. Robin Nusslock at Northwestern University, and he has what's called a brain map data set, which has 256 participants, over 200 of which have over 40 minutes of low motion data, which is kind of our threshold for where we think we can accurately define variance. And so at time one is assessed via structured clinical interview. There were 40 with lifetime depressive disorder, 36 with a lifetime anxiety disorder, 38 with a comorbid depression anxiety diagnosis, and also 126 that met no lifetime criteria for any disorder. So there's a good variability here in terms of clinical symptomology and healthy controls. And the thing about this data set, though, however, is that it's collected mostly with task data. So at time point one, we have 65.6 minutes total of data, but only 8.3 minutes of that is rest. So the rest of this 57.3 minutes comes from a combination of a couple fear paradigms and also this reward monetary incentive delay task. And so people are scanned at time point one and then also at time point 4, 3 years later. And so based on our kind of previous analyses here, we are now planning on trying to define network variance here by combining this task and rest data together to give us enough data to have reliable estimates of where network variants occur. And also as part of this study, they have a tri-level model of internalizing disorders to try to get at heterogeneity of clinical symptoms. So this tri-level model is pretty interesting. It's a hierarchical model, which has a general distress factor, which is related to all of the other psychopathology measures, which are in the model. And then there's some intermediate ones, such as fears and anhedonia apprehension. So those are kind of intermediate factors in the hierarchy. And then there's more narrow factors, which load onto a very select few amount of symptoms, such as narrow depression, worry, social anxiety, and a few others as well. And so just to show you some kind of preliminary stuff we're looking at here. So the first thing we wanted to do is kind of establish that we could get reliable variance by combining this rest and task data. And so we get very similar reliability curves to what I've shown you before. So these are showing you subjects, healthy controls who have never had any psychiatric diagnosis, and also people who have had a diagnosis of major depressive disorder. And as you can see, similar to the other reliability curves, after about 40 minutes, and most of them end up above 0.8, although there's a couple of stragglers in each one who are a little bit lower than that. And one of the things I'm really interested in looking at in this data set are longitudinal changes in network variance. So essentially, because we have these 2 time points, which each collect a little bit over an hour of data, there's a subset of subjects who have enough data that you can make network variance separately at each time point and try to look longitudinally at changes in their network variance and look at how those vary with clinical symptoms. So for instance, here we have this individual who at their baseline has a singular opercular variant right here in their temporal parietal junction and also at their 3-year follow-up. And same thing for their somatomotor variant right here, but also there's some differences between the time points. So here at the baseline, there's no variant here, but there is one at the 3-year follow-up scan. And the same thing with the frontal parietal here, and also no longer here at the 3-year follow-up. So I'm very interested in looking at those longitudinal changes. And just to wrap up really quickly here before time expires. So I'm really interested in examining the relationships between network variant location, network assignment, and psychopathology in this data set. And also I want to look at how variants change longitudinally with changes in symptoms. And so just to wrap up everything here, so we find that network variants are pretty similar across states, but do show some state dependency, and that combining task and rest data appears to be a suitable strategy for identifying these network variants. And so in our opinion, this method can allow network variants to be defined in data sets with a small amount of resting state data. So I'd just like to thank all of you for coming to the talk. I'd also like to thank my lab here at Northwestern, the GRATTON lab, and all the members of that lab. So I'd like to thank Zach, Megan, Derek, Alexis, Deanna, Allie, Katerina, and of course, Monkey. And I also want to thank the Nuslock lab as well as the NIH for funding. So thank you everybody for coming.
Thank you, Brian, for that wonderful talk. Virtual round of applause for Brian. We have time for 1 or 2 questions if anyone wants to raise any.
I had a question about how you might think about applying this to kids and developmental data, since there is so much variation throughout maturation and development in childhood. So would you expect these variants to be consistent, or would you have to wait until there's adult-like neural networks to look at these types of network variants?
So I think we haven't looked at this ourselves, but so my understanding is I think there would probably be more variability in the networks before 25 about. We've only really looked at this a lot in people that are 25 or older, which is really after a lot of brain maturation occurs. But I do think still that it would be very informative to do so, even if you couldn't be totally sure exactly whether the changes were due to, you know, developmental changes that are kind of somewhat, I guess, not totally divorced from environment, but somewhat divorced from environment perhaps, and other ones, you know, versus things like psychiatric symptoms in adults, which are, you know, kind of, you can sort of be having less or more severe symptomology at any given time, but at least you would, you know, have a more environmental explanation for why variants might be changing. But I definitely think that's a really interesting avenue to go down with this. We just aren't aware of any data sets with adolescents that have enough data at each time point to do something like this.
So if you have, let's say, like our lab collects for like 4 to 12-year-olds, and we have a little bit over an hour's worth of data, but it's only like a 6-minute resting state scan, like 2 5-minute tasks of a few different tasks, would you be able to combine all of that to do this type of precision MRI analyses?
So we typically set a minimum at 30 to 40 minutes. We usually try to stick to 40. So if you can get to 40 minutes of low motion data, we think, you know, that you can pretty much do this with any combination of rest and task, assuming that you can get to that threshold.
Okay, cool. Thank you.
Any final quick question for Brian?
I have another question for you,
I-
Brian. Oh, go ahead. I asked one last time, Jean.
I just wondering how so now the way you define the network is find a seed and then define the network, right? And then compare the individual network to the group, to the I just wondering how different way to define network will that affect the variant you find. Just-
So yeah, so well, so I guess when we define network variance, we look at the spatial correlation of functional connectivity between a group and an individual, but we haven't actually done any network analyses yet. So this is just we're essentially at a functional connectivity matrix level. How similar are these functional connectivity matrices if you stretch them out into a vector and correlate them? So it is, I mean, it could be possible to, define the networks that the variance should assign to. I mean, you could use different group averages or different network templates. I'm not sure if that's quite answering the question. I'm sorry.
Oh, yeah, I think I misunderstand the method. So you didn't define any network. You just calculate the connectivity itself and then calculate the spatial correlation.
Yeah, so once we get the spatial correlation, then we go to network assignment based on a template, but we don't start off by assigning them to networks and then trying to define variance that way.
Okay, thanks.
Thank you.
Brian, do you have time for one more question from Athena?
Sure, yeah.
All right, last question. Athena, go ahead.
Yeah, so when you're talking about these network variants, I can think of a couple different ways that they could be sort of the metric that could be used. And I'm curious what your thoughts are. So are you looking at the number, or are you interested in the number of variants someone has, or the size of the variants, or where, like what those networks are from? Like I can imagine there are a couple different things of interest there. What is of most interest to you in terms of what you're looking for in variation with, especially transdiagnostically as it applies to mental illness and MDD?
Yeah, so that's a great question. And I've also thought about this a lot and am still working on it. So my kind of idea sort of currently as I'm working through this is more in line with kind of what I presented before about how there's these very different accounts from meta-analyses of what the connectivity profiles between these group level networks are. And one of the things that kind of suggests to me is that it might not totally like it might not matter in isolation, like where a variant occurs or what network it's ultimately assigned to. But for instance, like the sort of like an interaction between them, like this area that's typically in the frontal parietal network is now in the default mode network, and how would that alter, like, because you could think about how like that would kind of wreak havoc on group functional connectivity, right? If you sort of have people who have these regions where you're like, this is their frontal parietal network and it's not, it's really anti-correlated to it because in this person it's a default mode network. So those kind of like interactions between what you would typically expect and what the connectivity actually looks like are something that I'm really interested in looking at.
Thank you. That's very interesting.
Thank you.
All right. Well, I want to thank Brian one last time. That was a wonderful talk. Thank you for joining us today.
Yeah, thank you for having me. It was really enjoyable.
And thank you all for coming as well. I hope you all have a wonderful weekend.